Hello, my name is Juliana, and I was an intern at the DC office working on the biomedical innovation workstream this summer. My project at Margolis focused on how we can increase competition in the cell and gene therapy market. And this involved a variety of different project tasks, such as looking at how we might shape the regulatory pathway for these future cell and gene therapies, as well as what different approaches might be to scaling up the manufacturing for these therapies. I developed a particular interest in the regulatory aspects. And so for my personal project, I decided to center it on how we might streamline global approvals of cell and gene therapies so that we can improve access to them around the world. So a brief overview. Cell and gene therapies are a truly new and exciting treatment option. For diseases, and especially rare diseases that lack attention, we now have treatments and even cures that are offering incredible new hope. And so these are very new and especially novel and also incredibly complex. And so that means that regulatory agencies are still catching up with what requirements for approval there should be for this class of therapies, especially evidentiary requirements. And if we consider how many of these therapies that are in development, given that there are over 1,220 clinical trials happening around the world, the global regulatory space must prepare for a new wave of market approvals and applications. And one way that it might do that is by seeking regulatory convergence, which is discussing how different national regulatory agencies around the world can align their guidance and make their recommendations more similar. And that will help to align innovators' expectations and help them to just comply with those guidances. So at its most basic level, what is a cell and gene therapy? Cell therapies involve the transplantation of human cells to replace or repair damaged tissues and/or cells. And so a more simple cell therapy might look like a bone graft or skin replacement, whereas a more complex cell therapy could be a CAR T cell therapy that treats cancer. On the gene therapy side of things, a gene therapy involves the transplantation of normal genes into cells in the place of missing or defective ones. Gene therapies are often taking advantage of new viral vector technology. And here on the next slide, we can see how Luxturna is a gene therapy that uses a viral vector to deliver a working copy of the gene into the eye for a person with a rare inherited retinal disease. And so this has incredible value and has an eye-popping price of $425,000 per eye. So we can see why these innovative payment approaches that are part of the broader Margolis project will be especially relevant. For my project, I chose to specifically focus on these types of therapies where they are a single administration. After one dose, you have long-lasting, durable, curative results. You have restored normal function or far improved function. And so durable cell and gene therapies have many reasons that they should receive additional attention from regulators. And one of those reasons is that many of them address rare diseases. And because of that, because they address such a small population, we want to maximize the potential population that they might cover to make it a more attractive business case to commercialize them. And so if we look at the existing landscape, almost 1/2 of all approved cell and gene therapies in the US, EU, and Japan received an orphan drug designation, which is a set of regulatory incentives to investigate new treatments for rare diseases. In the US, all durable cell and gene therapies receive this designation that are approved and on the market today. So another key area for why cell and gene therapies are uniquely suited to tailored regulation is that they're on the cutting edge. And so this makes them great candidates for pathways that are tailored for therapies that provide a substantial improvement over existing therapies. So in the US, every durable cell and gene therapy that's on the market today also qualified for the breakthrough designation. And in the EU, they have the analogous prime or priority medicines designation or program. And in Japan, they have the Sakigake program, which is unique because it specifically targets cell and gene therapies and expedites their approval. So given that kind of activity in the space currently, here are some different areas that we can look at for how we can improve the evidentiary requirements for these accelerated approval pathways, and just kind of streamline guidance that's out there. Specifically focused on approval. There are a variety of other considerations that one might take into account on how we can improve manufacturing standards, post-market approval follow-up from national regulatory agencies, et cetera. So area one is classification of solid gene therapies. And I'll give a broad overview of this just because there are a lot of different points to get through. So as I mentioned earlier, cell and gene therapies are not all the same level of complexity. A CAR T therapy has much different risk and just general understanding of that product as opposed to a bone graft. And so they have different stringency of requirements. However, there's need for further clarity on when a project is sort of on the edge and just, I would say, standardization across the globe. For example, in Europe, in some cases, different EU member states have different legislation. And so that means one therapy might have to meet more stringent requirements in one area than another, and that can make commercialization very challenging. Another kind of additional burden that comes in the EU and also in Japan is when cell and gene therapies, or specifically gene therapies, are classified as GMOs. And with that comes additional preclinical trial requirements, specifically evaluating the potential for the shedding of the viral vector into the environment. But scientific consensus shows that this is not a particularly high risk for gene therapy trials. And in the US, there's not this additional burden of preclinical trial requirements. And so literature kind of says that we should waive these requirements and have further discussion among agencies on how we can make research for innovators, especially for like multi-center clinical trials, easier to achieve around the world. A final consideration is regulatory exemptions. In the US and EU, they have these exemptions that carve out exceptions from regulatory scrutiny because they consider those therapies to really fall in the lowest tier of regulatory risk. If it happens in the same surgery, you're taking the cells out, they remain in the original form, and then putting them back into the patient, the FDA just considers that to be of incredibly low risk. However, there are opportunities to kind of take advantage of these types of exemptions that need to be discussed, and a variety of organizations, such as the American Association for Cell and Gene Therapies, have put forth recommendations for how we might better the utilization of these exemptions and make sure that every therapy has evaluation that matches the standard of risk for it. And so that's a theme in general with this first area. We want the classification of cell and gene therapies to match the level of evidence needed to kind of ameliorate the uncertainty surrounding that product. So going off of that, that leads us into area two, which is how we might standardize novel methods of data collection in clinical trials. And so one kind of novel method of data evaluation that's come into vogue is surrogate endpoints, which are essentially an indicator or a sign used to tell if a treatment work. And this is being used to approve many new cell and gene therapies. But in the FDA, we can see that they've been waffling on approving some of the new surrogate endpoints because each indicator needs to be validated. And in the EU, some payers are unwilling to pay these eye-popping high costs for therapies if it's not based on clinical outcomes, it's just based on surrogate endpoints. And so this is a topic that is in need of discussion around the world for how we can streamline their usage. In addition, the conditional approval pathway in Japan is an example of a country that's pursuing a really novel, exciting regulatory approach and something to look for other national regulatory agencies in the future. And so basically what Japan does to expedite the clinical trial process is they have a conditional approval pathway that allows for market entry after phase 2 clinical trials. And then they have post-market approval follow-up and additional trials that kind of occur concurrently afterwards. And so this is exciting because it makes access to these cell and gene therapies happen a lot faster, but at the same time, there has been some controversy in the scientific community about whether sufficient scientific evidence is grounding these approvals. And so this is something to look out for in the future and consider if there should be broader implementation of this. So finally, in area 3 is what we should do to increase cross-agency collaboration. After achieving further discourse and consensus around the topics outlined in area 1, outlined in areas 1 and 2, we can look to increase cross-agency collaboration and truly take advantage of that progress. And so given the orphan drug designation that I discussed earlier, they have a joint application between the EU and the US for innovators to apply for that designation. But that kind of activity could be expanded for more joint applications that for different kind of expedited approval pathways across more countries. In addition, we can consider work sharing. Work sharing is the concept that different regulatory agencies rely upon each other to help achieve their goals and fulfill their tasks. And so one example of that is the Mutual Recognition Agreement. And the MRA basically means that if the EU inspects a plant, the US will accept that inspection as if they had conducted it themselves. However, cell and gene therapies are specifically exempted from this agreement. And that is a key area where we could reduce the burden of inspecting these manufacturing plants and streamline their accreditation. And so essentially, my goal with my deliverable has been to highlight some of the different areas that are out there and that we can achieve regulatory convergence. And a lot of this has been based on what stakeholders has highlighted. And then once we achieve that regulatory convergence, how we can really, truly take value from that new knowledge and consensus and mobilize that within regulatory agencies. Thank you.